Okay, so today we're working on lab test 2 review. Alex, you don't need this pencil today?
No, I got last
That's good. So yes, today lab test 2 review, and so here we go. So exercise 5 is on enzymes, and we're supposed to identify reactants, products, and then I added that we need to also identify the enzyme in a chemical reaction. This is question number 1. So I wrote this chemical reaction right here. I picked this chemical reaction because it's the actual chemical reaction that we did in lab when we were working on enzymes. I found this chemical reaction pretty much on page 58 in your laboratory manual. You probably put a box around this chemical reaction on page 58 because it was what we used again in lab. So using page 58 in this chemical reaction, we're supposed to identify what are the reactants. So using the letter R, where should I put the letter R on this? On the left side, that's right. And in case you don't remember, this H2O2 is hydrogen peroxide. Hydrogen peroxide. And so hydrogen peroxide is the reactant. Okay, now we need the products. Where should we put the letter P for products? On the right side. And notice our products are both water and oxygen. These are our products. So both of these are our products. And then also I want you to be able to identify an enzyme in a chemical reaction. What's the enzyme here? Catalase. Very good. And I'm just going to use E for enzyme. So everybody okay on number 1, being able to identify reactants, products, and I added enzyme on a chemical reaction. Okay, super. Okay, number 2, I have defined enzyme, substrate, active site, and enzyme substrate complex. Please make sure you note page 57 for number 2. I'm going to have something like a picture from page 57. Now this isn't page 57, but this is something like page 57 because you wouldn't expect it to be exactly like what's in the book. So I just took this picture of an enzyme catalyzed chemical reaction. And so I want you to imagine if I gave you this picture but I took off all of the words. Took off all of the words, crossed out all the words. Could you label it if all the words were crossed out? And so the big problem to me is that I've noticed that students have is they sometimes can't tell the enzyme from the substrate. It's the only thing they seem to get messed up on is the enzyme and the substrate. So what are some visual clues to know which one's the enzyme? Say it again, Katie. It does not change shape. It doesn't change at all. Everybody check out the enzyme. Does not change through the whole thing. Okay, and what else? Any other comments? Okay, so Karen's saying it's kind of got some indentions in it. What do we call those indentions in the enzyme? The active site. And you would have to be able to identify the active site. So adding what Katie and Karen said is that if you'll note, the substrate changes into the products, but the enzyme never changes. And that the enzyme usually has some kinds of little curvature or indentions that are complementary in shape to the substrate. And those little indentions or curvatures are called the active site. Okay, again, so for number 2, make sure that you can label these diagrams if all the words were taken off. Okay, and then I have, what is the effect of an enzyme on the activation energy? Okay, first of all, activation energy, let me see one. Activation energy. So, let me go back. I'm going to draw a graph. This goes to the second part of number 2. And do you remember what capital E stands for? Energy. And then down here we're just going to do time goes by. And I'm going to do 2 different lines. I'll do the first one. Let me change colors. I'm going to do the first one in red. And then I want to do the second one. I'll do it in yellow. Everybody tell the difference between those two? You okay on the colors? Okay, so one of those chemical reactions is with an enzyme and one of them is not. Which one would be with the enzyme? The top one or the bottom one? The bottom one. How do you know the bottom one is using an enzyme? Because the lower energy of activation. So this bottom one is with the enzyme and the top one is not. So that just brings me to a discussion of what this little hump is. So I'm going to kind of dash this little hump in here. And we have two different humps. We have the top hump, which is bigger, than the bottom hump, which is smaller. And these humps are called the energy of activation. So let's go back to the question. What is the effect of an enzyme on the energy of activation? What does an enzyme do to the energy of activation? Excellent. It lowers it. So the answer is it lowers it. Okay, there's quite a bit to number 2. Make sure that you can look at the pictures on page 57 and label those pictures if you weren't given the words, and that you also understand the graph that we just drew and that enzymes lower the energy of activation. Okay, on number 2. Okay, super. I have question number 3. What happens when an enzyme is denatured? It stops working. And does anybody know why? What does that mean to be denatured?
It changes shape and they can't accept the substrate.
That's good. Changes shape, it can't accept the substrate. And I heard something else over here. And the enzyme then will slow down. That's very good. Okay, so it stops working because it changes shape. And you guys remember the shape of an enzyme will dictate whether the active site can receive the substrate or not. So let me write that down for you. It stops working because it changes shape. Shape is very important for an enzyme to work properly. Okay, second part of number 3. What conditions might cause an enzyme to denature?
High temperature.
High temperature. Good, Stephania. High temp. What else? Sorry, is that you, Emily? Okay, heat, which is high temperature, and then what else? Okay. What about low pH? Yes? What about high pH? Yes. Most enzymes work at a pH near neutral. It's rare to find an enzyme that can work at extreme pHs. So generally, extreme pHs will denature an enzyme. But okay, on question number 3. This is question number 3. Super. Question number 4. Well, we really already kind of looked at this. Question number 4, what is the function of catalase? Everyone, page 58 would be our reference page for the function of catalase. So, just using our words, what seems to be the function of catalase? Yeah, it speeds the breakdown of what? Hydrogen peroxide. Very good. Who's that, Aldo? Very good. Speeds the breakdown of hydrogen peroxide. Don't like that. And I'm just using the formula of hydrogen peroxide here, H2O2. Speeds the breakdown of hydrogen peroxide. How did we judge if this was happening? What did we look for? Bubbles. Excellent job, bubbles. And what was the chemical identity of the bubbles? Oxygen. Good, Alex. O2 bubbles. We looked for O2 bubbles. We were looking for bubbles. Questions about number 4. Take a look at number 4. Anybody number 4? Super. Okay, number 5. I actually want us to look at our data for number 5, and our data is on page 60. So please turn in your lab manuals to page 60. I have number 5. What effects do different temperatures have on catalase activity? And we had 3 different temperatures there. A refrigerator, which was somewhere around 4 degrees Celsius, a warm water bath or incubator somewhere around 40 degrees Celsius, and then a really hot water bath around 90 degrees Celsius. And that should be the data you have for temperature on page 60. Is that true? Around 4 degrees Celsius, 40 degrees Celsius, and 90 degrees Celsius? Yes? Okay. All right, so how well did our catalase bubble when it was cold? Did you guys get some good bubbling there? Yes? Okay, so it was good. How about when it was warm? Nice bubbling there. How about when it was hot? No bubbling. Okay, no bubbling. Really bad bubbling. Why didn't it bubble when the catalase was so hot? It denatures. Good. So the really high temperature, like you guys said a few minutes ago, the really high temperature will shake the enzyme so bad that its bonding will break and it'll literally lose its shape and fall apart. But okay on that one. And our data support that idea. So please make sure you look at your data on page 60. It goes with number 5. Okay, number 6 is omit. We didn't have time to do that activity, so number 6 is omit. So that's why I jumped over to number 7. What's the effect of a pH outside the optimum range of an enzyme? Okay, well, let's check our data and let's see the answer. So this data, everyone, should be on page 62. Page 62. Okay, what is the effect of a pH outside the optimum range of an enzyme? Well, let's look. Let's see, test tube 1, correct me if I'm wrong. Did you get a pH of 1? Yeah, it was pretty acidic, don't you agree? Did we get good bubbling on test tube number 1 at the bottom of page 62? Did we get good bubbling there? No. Okay, test tube 2 was somewhere around 7, maybe 6, 7, or 8. Is that true? Did we get good bubbling there? Indeed, yes we did. Okay, and then test tube number 3 had a pH, I'm just guessing, it was either 13 or 14. About 14. That was super basic. Yeah, very high on the base. Okay, so what do you think the effect of a pH outside the optimum range of an enzyme would be? Would you go and say that the pH, high pH or low pH would speed up an enzyme? No. What would you say? Slows it down. As a matter of fact, it looks like it stopped it. Yeah, it stopped it. Yeah, stops the enzyme. And earlier, you recall, Emily told us really low pHs or really high pHs will denature an enzyme. Okay, so to make sure everybody's clear, it says what would acid, this is acid, everybody, HCl, that's an acid, or what would base, NaOH, that's a base, what would acid or base do to catalase? It starts with the letter D. What would it do to catalase? It denatures it. Good. And again, our data support that because the acid in test tube 1 stopped the enzyme from working, and the base in test tube 3 stopped the enzyme from working. So I drew those test tubes in this next slide. And this first one, this is supposed to be test tube number 1. This one's test tube number 2. This one's test tube number 3. And just look at your data one more time. I want to put bubbles in these test tubes. Should I put any bubbles in test tube 1? No bubbles. How about test tube 2? Yeah, just lots and lots of, this one bubbled very well. And then what about test tube 3? No. Now, in this drawing, I'd like to also add in the pH. The pH. What was the pH for test tube number 1? 1. pH of 1. How about test tube number 2? Around 7. Do you agree with Alex? Around 7? Yes? Okay. And then lastly, test tube number 3, pH? 14. Now, guys, all I did on this visual is I just drew out those 3 test tubes that you guys have on page 62. We have it with words there, what was in test tube 1, what was in test tube 2, what was in test tube 3. I just drew it out so that you could just lock in on a visual of what was in each test tube. Check out that acid was in test tube 1, water was in test tube 2, and base was in test tube 3. And every test tube was the same on catalase and hydrogen peroxide. Because you guys have learned by studying experiments that you only can vary one thing during an experiment, just one thing, and that everything else has to stay the same. So if you'll notice, catalase was in each test tube, and then hydrogen peroxide was in each test tube, but what was different is the acid in 1, the water in 2, and the base in number 3. So we're okay with my visual to go along with number 7. Number 7. Okay, looking back at questions 1 through 7, does anybody have a question about any of those? Is it okay? Celeste, do you need a review sheet? You got it on it? Okay. Okay, well, super. Then we're going to roll over to exercise number 6 on photosynthesis, page 67. Okay, so photosynthesis, we have to know the reactants and products of photosynthesis. Your review sheet says know the raw materials. Whatever. Those are called reactants. So I just went with the actual word because, you know, we are in college, so we can use bigger words. Reactants and products of photosynthesis. So I hope that you start or put a box around the chemical reaction on page 67. You have to know the chemical reaction of photosynthesis. Buddy, check it out that we have, I'll go ahead and write it. We have CO2 as a reactant, and we have water as a reactant. And you guys know that you have to have some sunlight or solar energy. Or a light bulb will work. You can do it with a light bulb. And then you make this thing that's called CH2O. Does anybody know what CH2O is supposed to be? Carbohydrate. Who told me that? Very good. Isabel said carbohydrate. And, of course, that's what the plant is doing this to make, is carbohydrate. That's what's going for, is to make that carbohydrate. And then it makes this very useful waste product called oxygen. Very nice. Thank you very much, plants. Okay. But, okay, on the chemical reaction, overall chemical reaction for photosynthesis. Okay, then the next question was, in what organelle does this process occur? Chloroplast. Very good. And there's an excellent drawing of a chloroplast on page 67. Page 67. So you should be able to spot that as a chloroplast. Chloroplasts are easy to spot. They're kind of oval shaped for most pictures. They have these internal membranes called thylakoids. So they're kind of easy to spot. They're nice sized organelles. All right, next page. Question number 2 is paper chromatography, page 68. So page 68 is a great reference for number 2. I have, what solvent is used in paper chromatography? Well, for our paper chromatography, the solvent that we used was a nonpolar solvent. So that's really what's important, is it was nonpolar. So was it water? No. Why? How do you know that? Because water is polar. This was, you don't have to memorize this, but it was ether and acetone. Some of you are familiar with solvents. You know, maybe your parents have them at the house. They keep them in cans all closed up because they evaporate real easily. And nonpolar solvents are great for dissolving things that are nonpolar. And the pigments in a leaf are nonpolar, so that's why we used a nonpolar solvent. Okay, how does paper chromatography work? Everybody check it out on page 68. You may have written a sentence. Paper chromatography separates molecules. That's what it does. It makes things separate away from each other. And then it separates molecules based on a certain chemical criteria. What criteria does it separate molecules based on? Solubility, that's right. So I'm going to write that down, that it separates molecules. And in this case, we did the pigments in a leaf. And it separates these molecules based on solubility, how well they dissolve in the solvent. If you were working with water-soluble pigments, you could use water as a solvent. But we were working with water-insoluble pigments. They were nonpolar. They wouldn't dissolve in water. That's why we had to use the nonpolar solvent. Okay, so this is my little drawing of what we got. Some of us did really well that day, and some of us not so much. And so, and some of it had to do with our mashing ability, and some of it had to do with our scissor ability. I remember that too. Yeah, that was an interesting day. But anyway, here's a paper chromatogram that might be what you would see on a test. Just showing it to you. And then you would have to pick out which pigment would be in which spot. Don't forget, this is a multiple choice test. So you just have to memorize who's at the top, who's in the middle, and who's at the bottom. Okay, so looking at page 68, letter C, what would be this very top pigment right here? Go ahead, this yellow one right here? Carotene. It is carotene. And then you come down, and there was another yellow one. What would that be? It's xanthophyll. Okay, then there was a green one, and there was quite a bit of this green one. What is it? Chlorophyll A. Good. And then most of us were able to find this, although a few of us had some difficulty finding this last one. What was it? This one's chlorophyll B. So you are going to have to memorize who was on the top, who was in the middle, who was at the bottom. And then you will have to answer this question. Given these four choices, which of these four is the most nonpolar? It is carotene. Who told me that? Stephania. She's right. This one was the most nonpolar because it went the farthest. That means it dissolved the best in the nonpolar solvent and moved the farthest. So the most nonpolar was carotene. Is okay on that one? Paper chromatography? Great. Okay, now as we're going to page 72, I just want to draw your attention to page 70 as we're moving over in our book. Just take a quick look at page 70. I just want to remind you that the light coming from the sun is white light, and white light is actually made of all the colors of the rainbow. Just want to remind you about that. Okay, then we're moving on over to page 72. Page 72. Guys, let's study the graph on page 72. And let's just specifically look at chlorophyll A, because you guys have learned chlorophyll A is the essential pigment of photosynthesis. So let's just look at that one, the graph for chlorophyll A. It says, what colors of light does chlorophyll A absorb the best? Purple. Okay, got some blue. Anything else? Orange and red. I agree. To figure out what that absorbs the best, what do you look for? Yeah, to figure out if it absorbs the best, what do you look for? Peaks or valleys? Peaks. And the reason you look for peaks is look at what the y-axis is. What is the y-axis? Absorbance. So if it's absorbing, you'll get a peak. Does everybody see how that makes sense? Super. Okay, what color or colors of light does chlorophyll absorb the least? Green. A little bit in the yellow. And how do you know that? Yeah, it's low. Absorption is low. So there we look for the valleys, correct? To figure out what a pigment absorbs, we look for peaks. To figure out what it's not absorbing, we look for valleys. Now, does this make sense that chlorophyll looks green to us? Okay, everybody turn around and look out the window. Everybody see all that green out there? How come you're seeing that green? It's reflecting green. Chlorophyll's reflecting green, and that's what our eyes perceive as what's being reflected, not absorbed. But okay, on why plants look green? It's great. Now, still on page 72. Still on page 72. Why is it beneficial to have several different plant pigments involved in photosynthesis? This is what they mean. Look at that graph again. It's not just chlorophyll A on that graph. There's also chlorophyll B. There's also the carotenoids. And we know plants have all of those pigments in their leaves. So why is it plants have all those pigments in their leaves? Good. Now, if you can absorb more colors of light, you get more energy, because light is energy. Don't forget, capital E is energy. My abbreviation for energy is capital E. So the more colors of light you can absorb, the more energy you're absorbing. So it makes you more efficient. Okay, second question on number 4. Given our answer that Stephanie gave us, what color of light is the best color of light for photosynthesis?
White.
Very good. Who told me that? Is that Stephanie? She's on the fire today. White. Does everybody understand why Stephanie picked white? Why did Stephanie pick white? Thank you, Tim. It's all the colors. It's all the colors. So that makes sense. Okay, what about what color of light is the worst? Black. Black is an absence of light. We gotta have some light, people. Who said green? Green's correct. Is that Emily being quiet? Dayanara? Who's being quiet over there? Emily? How come green would be the worst color of light for photosynthesis? Tell me again. Plants don't absorb it. Is that Alexander? He said plants can't absorb green. You just said that, class. You just said plants reflect green. They don't absorb it. So green would be the worst color. You guys know black is the absence of light, right? Like when we turn off the light in a room, what color do we get? Black. Okay, good. True or false? Most plants carry out photosynthesis only during the day. That's true.
Wait, I have a question.
Yes.
So if black is the absence of light, is the shirt very black or no?
Nothing's being reflected. So your the black pigment in your shirt is absorbing every single color, so nothing is being reflected. So it is the absence of light hitting my eye. I can't perceive any light coming off of your shirt. Jordan? Okay, say it slowly.
It was on the lecture thing, like that question actually was hard, like how, like talk about some plants are visible. It was on the
Lecture test?
yeah.
I'll have to look. It's a good question.
I just put all of them.
Picked all of them? speaking of that, since Jordan brought it up, anytime you want to come and look at your lecture test and look at questions that you were wondering, did I get that right or did I get that wrong, and kind of learn from your test, you're always welcome to come to my office and do that. Just let me know when you're coming so I can make sure and be there. And that way we can discuss questions you may have had during the test. Okay, so everybody's okay that most plants carry out photosynthesis only during the day. And the reason is this part of photosynthesis. Does anybody know what photo of photosynthesis means? Light. That's what photo of photosynthesis means. It's very good. Okay, try the second one. True or false? Plants carry out cellular respiration only at night. First of all, do plants do cellular respiration? Every living cell does. Okay, so true or false? Plants carry out cellular respiration only at night. That's false. Plants carry out cellular respiration every day, every minute, every night, every minute. When cells stop respiring, they die. So this is a 24/7 thing. Respiration has to occur all the time for every living cell. It doesn't matter if you're a plant, an animal, bacteria. I don't care who you are. You're doing cellular respiration every day, every night, all the time. But okay, on this one's false. Super. Okay, number 6. In our notes for number 6, everyone, if you would, page 74, we have a few notes. I think we wrote by hand on page 74. I have number 6. Why did the phenol red turn yellow when carbon dioxide was added? Well, in case you didn't remember this one, this is where you had a beaker of water and we added phenol red to it, and then I gave you a straw. This is the day we had to blow into the water with the straw. Somebody at your table had to do it for you. I don't know who that lucky person was. But if you remember, they blew, it was Tim at his table. He remembers. He had to blow with the straw into the beaker, and the red color went away, and instead it turned yellow. Now there's a chemical reaction that you maybe wrote down that explains why that happened. Do you guys have that chemical reaction? Okay, so there was water in the beaker, and what did you blow through the straw? CO2. And that combines together because CO2 is very soluble and reactive with water. And that all combines. If you just add that all up, slap it together, you get H2CO3. If you just count the atoms, you can see that's what it adds up to. And H2CO3 is carbonic acid, an acid. It's a very weak acid, but it's still carbonic acid. And that's why the phenol red turned yellow. Tim?
On my notes I put the double arrow.
Yes. I was going to add that next. So very good. So that day when you're pushing all that CO2 in, you're pushing it to the right. But Tim is correct. This is a reversible reaction and it can go back the other way. Tim is correct. When we're pushing that CO2 into the beaker, you were pushing that chemical reaction forward, but then after you stopped doing that, Tim is right, then it can reverse itself. So we made acid in the water is the basic idea, and that acid is due to your carbon dioxide in your breath. Okay, so I got myself a little drawing there. Check it out. Here's your beaker. That's a little plant we put into the beaker, and we put it in the window. Now, when we left it, that beaker wasn't pink, it was yellow. But when we came back 5 days later, it was pink. And did you guys write that down on page 74? When you came back, it was pink? Okay, so I made it pink here. This was after 5 days. I have, why did the solution turn back to red, also known as pink, after being placed in the light? So what did you write down? Why did it turn back? Plant did photosynthesis. And what gas did it use? CO2. I want to abbreviate photosynthesis there. So that's why Tim was pointing out the reversible arrow there. So you pumped a bunch of CO2 into the water with your breath, and then the plant used it up doing photosynthesis. And so he's right, it reversed the arrow back, and the plant took up the CO2. Okay, on this part. Yes. Okay, why did the solution stay yellow when we placed it in the dark? That's supposed to be a moon, in case you don't know what I'm drawing there. We're in the dark here. And we put the beaker in a drawer, but I can't draw a drawer with a beaker in it, so I just put the moon there. This is supposed to replicate or simulate, the darkness. And notice it's yellow. It stayed yellow. So why did it stay yellow? Excellent, Alexander. No light, no photosynthesis. Everybody's okay on that one? Super. Okay, number 7. Why is it important to have autotrophs? They can make their own food. Heterotrophs, they can't. And decomposers in an ecosystem. So check out my ecosystem. It's like my aquarium in the back, right? Check it out. I got my water, I got my fish, I got my plants, I got a few plants back there. And then check out the bottom, I got some gravel in the bottom. Do you guys know why you're supposed to put gravel in the bottom of an aquarium? It provides surface area for bacteria. Surface area for bacteria. Why do you need bacteria in an ecosystem? To chemical recycle, to decompose. They're your decomposers. Yes, sir?
The heterotroph eat the autotroph's food, it dies, decomposes, recycles.
Okay. So what Jordan is saying is the heterotroph can eat the autotroph, and then any waste products made by the heterotroph or any waste products produced by the autotroph can be digested by the decomposers. If you don't like my picture, page 74 is a pretty good picture. Check out the corn there, and there's a corn patch and a cow. Now, Jordan's pointing out the food cycle that's going on there. Right, check out the food coming from the corn. I know cows like corn. Yeah. And then cows will give off waste products, CO2 and water, that the corn likes. Everybody see that cycle going on there? But there's another cycle of gases going on there. What gas is the plant giving off? Oxygen. And notice the cow's using the oxygen. And then what gas is the cow giving off? CO2. And everybody notice the corn using the CO2. Okay, so everybody see we've got a cycle there. So having autotrophs making food and oxygen is good for heterotrophs. Having heterotrophs making water and CO2 is good for autotrophs. Okay, now, do you have to have decomposers? Yes. If you didn't have decomposers, then what would happen? Be a buildup of waste products. And you wouldn't have any of the inorganic nutrients in the soil or the sediments in my aquarium that the autotrophs need. Okay, I got one more thing. I don't know if you've looked at my aquarium, but it's a little bit open in the back. That's where the little waterfall from the filter is bringing oxygenated air into it. But what if it was completely closed? So I'm going to put a board on top of it. Would that necessarily mean the death of my autotrophs and heterotrophs? No. Why? Because I still have a cycle going on. Okay, so the heterotrophs, they're producing CO2 that the autotrophs need, and the autotroph is producing O2 that the heterotroph needs. But would my aquarium last as long? No. So everybody agree that little board, if I put the board on there, the aquarium could last for quite a while longer, but having an influx of gases from the atmosphere helps the ecosystem do even better. Yes? Everybody's okay with that? So look over number 7 one more time and make sure that you're okay with the idea of why ecosystems need all 3 of those things. Okay, and then there's just one more thing this ecosystem needs. Okay, look at my picture. There is something missing. Thank you very much. It needs the sun. Why? Why does it need the sun? Why does this ecosystem need the sun? For the autotrophs. Because the cycle will stop if the autotroph cannot make its food and oxygen. But I agree we need the sun. Super. All right, so we're going to leave that. Photosynthesis, we're going to cellular respiration, page 77. I need the reactants and products of both aerobic and anaerobic cellular respiration. Okay, on page 77, you tell me there's a chemical equation there on page 77. Is that aerobic or is that anaerobic? That's definitely aerobic. Okay, so I wrote that one down for you here, and it's also page 77. I've got glucose and oxygen makes carbon dioxide, water, and ATP. Now, does anybody remember how many ATP it makes? 38. That's good. Is that you, Alexander? 38. 38 ATP. This one's aerobic. Aerobic. How do you know this one's aerobic? Because of the oxygen. Very good. Because it's got the oxygen that makes it aerobic. Okay, and this is on page 77 in your lab manual. Page 77. That's where I got this one from. Okay, so I'm going to go to the next one. This one's on page 78. Page 78. Everybody check out page 78. Okay, now this one makes carbon dioxide from glucose. It makes ethanol, which is what, C2H5OH. Oops. My H didn't come out. That's ethanol, drinking alcohol. And then it also makes ATP. And it went ahead and gave us the answer of how many. This one actually told us 2. What do you think about this one? Is this one aerobic or anaerobic? How do you know it's anaerobic? Because it doesn't have oxygen. So this one is anaerobic. And I got this one off at the top of page 78 in your lab manual. Page 78. You guys, you don't have to write this out on a test. You just have to recognize which one's which. Okay, remember it's a multiple choice test. You just have to figure out which one's which. Is it okay on that one? Okay, let's look at question number 2. It says, both anaerobic and aerobic cellular respiration ordinarily begin with what molecule? C6H12O6, also known as glucose. Everybody agree with Tim and company? They agree it's glucose? Okay, great. And then, yes, you're going to have to know how much ATP each one makes. We wrote that down already. 38 for aerobic and 2 for anaerobic. Okay, you have the same 2 choices, anaerobic or aerobic. In which do you think the glucose is completely broken apart? The one with the oxygen, which is aerobic. Here's how I know. I know glucose is for energy. Everybody knows when you have glucose in your body, you're using it for energy? Uh-oh. Sorry, guys. There it is. We back? Yes. Does everybody know that glucose is for energy? Okay, so which one of those chemical reactions did we get the most energy out of? The aerobic. So we must have chopped the glucose up completely to get all that energy. We got hardly any energy at all from anaerobic, so we must have not gotten much broken away from the glucose. So how you would know is the energy content. We got way more energy out of aerobic, so that must be the one that glucose is completely breaks down. And again, you have to know the ATP yield for both, so make sure you know those. Okay, the fermentation experiment. I did my little drawing here, and if you don't remember this one, this is where we had itty-bitty test tubes that we put sugar and yeast in, and then we put them inside of another test tube, and we had to do the flippy thing. And I found the data for this on page 82. Page 82. So I just looked at the data and drew some test tubes. You can tell test tube number 1, the sugar in there was glucose. I think you wrote it down on page 82. And in test tube number 2, the sugar in there was fructose. Test tube number 3 was sucrose. And test tube number 4 had no sugar at all. It was water. And then every test tube got yeast. And then can you tell, guys, that there's some gas in some of these? Okay. All right, so this is what I'm referring to on number 4, the fermentation experiments. So here we go with the questions. Did the gas bubble increase or decrease with each of the solutions used? Now let's look at our class data. Did we get some nice gas production in tube number 1? Yes. How about tube number 2? Yes. Tube number 3? Yes. Tube number 4? Not too much. How come we didn't get much gas production in tube number 4? Can yeast use water for energy? No. Can anything use water for energy? Any biological system, any cell? No. Okay, so what was the purpose of number 4? That was our negative control. Very good. So we were showing you had to have some kind of organic compound, a sugar, for the yeast to do anaerobic cellular respiration. Okay, so that was our experiment. Now, in this experiment, I want you to be able to tell me what the gas was. So did the gas bubble increase or decrease with each? So it increased with 1, 2, and 3, but it did nothing. It did nothing for test tube 4. That was our negative control. So I'll just write that down, control. And then I put cells cannot use water for energy. It's a really messy E. I'll try again. A little better. Then the last most important thing is, what was that gas that they were making? Tell me again. It is. Thank you, Karen. You're being chicken back there, huh? CO2. So I'm just going to back up for a second while you're writing that down. I'm going to go back to this chemical reaction. But you see the CO2 as a product? And then look at this one. See the CO2 as a product? So CO2 is a product of cellular respiration. CO2 is a product of cellular respiration. Okay, the aerobic cellular respiration experiment. All right, I couldn't draw this. Sorry, guys. This is just a little beyond my expertise on drawing. So I made it for you to remind you what it looked like. Do you guys remember this one? Okay, so we have a ring stand with a clamp, a big test tube, and then we have this pipette that's been stuck in this stopper. And you may remember we put cotton in the bottom with some CO2 absorber called KOH. And then we had some dry cotton, and we put some germinating peas in there. Okay, so this is the experiment I'm talking about. What did we do with the shaving cream? Yeah, so we plugged up the end, and then we used it to measure how much oxygen the seeds were using up. How did we keep the seeds from doing photosynthesis? Aluminum foil. That's very good. We wrapped them in foil so that they would be in darkness. So everybody remembers that experiment. Now, if you didn't remember that, I just want to reference page 79 is a pretty good page to look at. Just remember the 3 test tubes. Test tube 1 had the germinating peas with the cotton. Test tube 2 had the dry peas with some glass beads. And test tube 3 just had glass beads. Okay, then we put our data on page 80. Let's check out our data. Look at our class data for this one. Which test tube, 1, 2, or 3, had the most movement of the shaving cream? 1. And that's because the cells are the most metabolically active, so they were doing the most cellular respiration. So that's just a reminder of what that looked like. So you might want to put in your notes to look at page 79 and 80. For number 5, page 79 and 80. Let's try number 6. Which direction did you expect the shaving cream to move during the aerobic cellular respiration experiment? In or out? In. And why? What are they using? They're using oxygen. Kind of put together what you guys just told me into a sentence. Cells using oxygen in cellular respiration. So we've already answered question 7. But check out question 7. Did we already answer it? It says, what was the gas taken up by the germinating peas? It's oxygen. Everybody agree we already answered that question? Okay.
I have a question.
Yes, ma'am.
If there's like pills that say it helps with cellular respiration, what does that mean?
It's speeding up your metabolism. These pills are probably caffeine pills. Right now you can sell caffeine pills legally. FDA will let you sell caffeine pills to speed up your metabolism. Caffeine speeds your metabolism. You got to be careful though, it'll dehydrate you also, and it'll make your heart race. Okay, good question, Stephanie. Anybody have a question about exercise 7? All right, super. We're going to move to mitosis now, page 85. Okay, now guys, you're going to have to know what happens in the stages of mitosis, and I just made a list here of parts of the cell cycle and stages of mitosis. I want you to go back into either your lecture notes or your textbook or your lab manual, whatever you want to go back and review the stages of mitosis. Just want to mention a really nice reference page for this entire exercise is page 88 and 89. If you'll just glance, that's the page that has not only really good visuals, but it also has some words that describe each of the phases. So if you'll review page 88 and 89 of mitosis, that would be a good reference page for mitosis, no matter if you're studying for lecture or lab.
So are we going to need to know like the words or like what it looks like?
Very good, Nikki. We're going to have to know what it looks like. And Nikki's right, the words will be on the lecture test. Very good point. Okay, I took a picture of the cell cycle. Now this actually is a picture of the cell cycle from the textbook because I was telling you I like this cell cycle better. Anybody spot what's different compared to page 86 in your lab manual, your cell cycle, your lab manual cell cycle, and the one I took from the textbook?
It shows 2 cells leaving the cell cycle, and in yours it shows 1 re-entering.
No, I got 2 cells, but they're just coming off the bottom.
Well, yeah.
There's something else.
Oh, G0?
There you go. Yeah, I like that. That G0. That's why I picked this picture, because I like it better with this G0, just a little out pocket of G1. But anyway, on page 86, I think you wrote down what happens in G1, S, G2. So I'm just going to throw something out there. What stage of the cell cycle are you in if the DNA is replicating? S. Very good. What stage of the cell cycle are you in if chromosomes are lined up single file on the equator? Good, Karen. Metaphase. Yeah. What stage of the cell cycle are you in if the cell is greatly increasing in size? You're in G1. That's where it really grows a lot. Okay, what stage of the cell cycle are you in if you see cleavage furrow formation? Telophase, cytokinesis. That's very good. All right, now everybody look at the second part of number 1. Know the form of DNA in each stage. Okay, what I mean by that is it chromatin or is it chromosomes? So let's talk about the difference. Chromatin is loosely dispersed, and chromosomes are tightly coiled. So we're going to start with G1. Everybody look at G1. If you're in G1, is your DNA tightly coiled up, chromosomes, or is it loosely dispersed, chromatin? It's loosely dispersed. So I'm just going to kind of draw it up here, just all loosely dispersed. Can't really see that very well. Let me make it darker. Try again. Oh, yeah, that shows it better. Okay, how about S? Still loosely dispersed. But everybody check out how I'm going to draw it differently this time. Look how I drew it in G1. It's just one-sided. And then now look at it in G2. It's double-sided. Why? DNA replication. Alex is right. Everybody see how now it's got two sides to it because it copied itself. Everybody see how I drew that? Okay, what would it be in G2? Still loosely dispersed. Would it be double-sided or single-sided? Double. It would be double. Now what do we call this loosely dispersed DNA? Chromatin. This is called chromatin. 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 It's loosely dispersed DNA. All right, now we're going to go into mitosis. Is the DNA loosely dispersed or tightly coiled during mitosis? It's tightly coiled. So look how I'm going to make it really nice and thick. And check out it's double-sided. You guys see this okay? Prometaphase is off the books. We're not doing prometaphase. Okay, metaphase is going to look the same. Nice and thick, double-sided. This is my drawing for metaphase. But what about anaphase? They pull apart. So instead of being double-sided, now it's going to be single-sided. And the same thing on telophase. Everybody check it out. Notice here in anaphase and telophase that the chromosome is single-sided. And then look, we go back to G1 and notice your chromosome is single-sided. And instead of being tightly coiled, now it's loosely dispersed. So everybody can see we kind of got a cycle going here. Single-sided till you get to S, putting a star there. And then double-sided till you get to anaphase, and I'm putting a star there. Why did I star S? That's where DNA replicates. So you went from single-sided to double-sided. Why did I star anaphase? It's when you went back to single-sided. So I starred the changes in whether you're single-sided or double-sided. Okay, when I drew my DNA all long and thin, DNA was chromatin. When I drew my DNA real tight and thick, DNA was chromosomes. Yes, Tim?
What kind of DNA does mitosis have?
Say it again.
What kind of DNA does mitosis have?
Chromosomes. Okay, so let's be clear about that. Mitosis has chromosomes. Interphase has chromatin. It's a good question, Tim. Mitosis has chromosomes. Interphase has chromatin. Okay, so that just brings me to this slide. We've kind of stressed this already. So chromatin is loosely dispersed and it's found during interphase. I'm just going to write it down for you for those of you that are going to look at this on YouTube. And this is found during mitosis. Okay, picture cell cycle again. All right, number 2. Okay, there's a whole bunch of words here that I could ask you about in either drawings like this or just with some multiple choice questions. I've got these chromosomes drawn up here. Everybody can tell it's a chromosome. I mean, can't you tell? Why are there 2 of them? Yeah, these are homologous chromosomes. We could pretend the red one came from our mom and the blue one came from our dad. Okay, now, every chromosome's got 2 sides. What do you call one side? A chromatid. So I'm going to go ahead and draw that arrow right there. A chromatid would be one side. And then what do you call a centromere? The center where they're attached to each other. Remember, one chromatid is identical to the other chromatid. Okay, you're okay on the parts of a chromosome. We did this in our lab manual, page 86. If you'd like a picture from your lab manuals, page 86. Okay, still on this question of definitions, what is cytokinesis? If you had to just go up and tell your 3rd grade cousin, what is cytokinesis? What would you tell them?
It's when a cell comes to.
So you're dividing something. What are you dividing? It's division of the cytoplasm. That's good, Jordan. Division of cytoplasm.
I don't think the 3rd grader would know what cytoplasm is.
Okay, 7th grader. Let's say it was a 7th grader. Does that sound better? Did you know cytoplasm in 7th grade?
I didn't know the spelling, but I knew what it was.
About 7th grade? Was it 10th grade? I don't know where you went to high school. Was it 10th grade? I don't know. All right, spindle fibers. If you had to tell a 10th grader what spindle fibers do, what do? Pull. I like that, Alexander. They pull what apart?
Sister chromatid.
They pull sister chromatids apart. Okay, then lastly, centrioles. Who has them? Animals. There's 1 pair per cell. Do we even know what they do? We're not real sure. They're not real sure. They have something to do with cilia and flagella. We know that, but we're not really sure what they do. They're in the center of the centrosome, which makes spindle fibers. But we don't think that they're making the spindle fibers because plants can make spindle fibers and they don't have centrioles. Okay. So I have number 3. What are the differences between plant and animal mitosis? Any ideas?
In animals, mitosis, there's things for
Good.
And I've got cell plates.
Good. Like both of those.
someone over here is the second.
Yep. And then the second thing was that only animals have centrioles. Plants do not. And guys, that's about it. Okay, so it's pretty similar, plants versus animals. That's just about it. How they divide their cytoplasm and whether they have centrioles or not, that's about it. Everybody okay on that one? Okay, so I just drew the 2 cells here. Can you tell me what, I have my arrow on here. That's a cell plate. So what kind of plant would that, excuse me, I just told you the answer. That's a plant, yeah. How about this one? Cleavage furrow, that would be an animal. Good. We're good on that. Super. Why is a cleavage furrow suitable for animals but not for plants? Very good. Plants have a cell wall. Tell me about that cell wall.
It's very sturdy.
Sturdy.
Turgid, was it?
Rigid. How about rigid? Let's use the word rigid. Yeah. So it won't be able to bend in and form a cleavage furrow. So it's the cell wall issue. Yeah. Plant cell wall is rigid. It can't bend in and make a cleavage furrow. So everybody's okay with that? That's why they can't do it.
So turgid means like loose?
Turgid means that they have a lot of pressure against their cell wall. It is a word we use to talk about plant cells. That is true. Okay. You have to know the stages of plant and animal mitosis in 3 ways. I remember telling you you're going to be tested in 3 weeks. One way is the pop bead chromosomes. And to study that, I would look at page 88, 89, through the middle of the page. Then you also have to be able to tell the stages of mitosis on plant and animal slides. And again, page 88 and 89, animals are across the top and plants are across the bottom. Guys, don't forget, you do not have to know pro-metaphase. We're just doing prophase, metaphase, anaphase, and telophase. Okay, and then lastly, models. Where are the models at? That side of the room. And if you wanted to take pictures of the models, feel free to do so, or just stand and look at them. But the models will be on the test also. Plant versus animal. Plant have the green cards, and animal have the blue cards over here on the side. So take a look at them before you go today. Guys, these are just pictures from your book, page 88 and 89, showing you my admiration for those pictures. They're great. Okay, I think we're on the back. So now we're going to move to meiosis. And guys, if you will, meiosis is well done in your lab manual, also page 96 and 97. If you would, flip over to page 96 and 97 for meiosis. Okay, what is diploid? What's DI mean? 2. Does anybody know the diploid number for humans? 46. What's the shorthand notation for diploid? 2N. Very good. I'm going to put that right there. 2N. Okay, on diploid? Okay, what's haploid? 1 set. Very good. And shorthand notation? N. Good job. Okay, then I have, what are homologous chromosomes? Well, first of all, they've got to be a pair. They're the same. How come you always get a pair of homologous chromosomes? Why are they always in pairs? One from each parent. Very good. One would be maternal from your mom, and one would be paternal from your dad. Everybody okay with that? Super. Everybody okay with these definitions here? There are stages of meiosis, page 96, 97. Okay, be familiar with all the pictures of gametogenesis. I found gametogenesis on page 101. You know, the basic thing I need you to know is, where do men do spermatogenesis? In the testes. And per original diploid cell that starts spermatogenesis, how many sperm can you get? 4. Okay, check out page 101 and see that at the top. Spermatogenesis occurs in the testes. Per original diploid cell that starts spermatogenesis, you get 4 sperm. But okay with that one. Okay, oogenesis at the bottom of page 101. Where does oogenesis occur in women? Ovaries. Per original diploid cell that starts oogenesis, how many functional ova or eggs can you get? One. And what are the other three things? Polar bodies. What happens to polar bodies? They die. Okay, yes, sir.
I don't know if this is going to be talked about later, but in karyotype, which is the smallest set of chromosomes?
21. Okay, page 102 is the ovary. So, what's up?
What did you say about polar bodies? You get one egg and then
3 polar bodies, and they die.
Okay.
Yes, Miss Anatomy 2, they do. So, page 102, ovary. I'm thinking it'd be nice for you to know an ovary from a testy. That's just me, okay? And in this class, we only get to see them microscopically. But in Biology 2 that you'll take next semester, you'll be able to see them inside the pigs that you dissect. You'll get to see the mammalian ovaries and the mammalian testes, so you can tell the difference that way. But anyway, on page 102, I want to have the ovary slide, so you can be able to tell me, by looking through the microscope, if I say name this organ, you have to pick ovary. And then check out that picture on page 102. There's a cell living in a big kind of C-shaped structure. That cell is the oocyte, the egg. And the big swollen structure it lives inside of is a follicle.
Is that what slide you're going to show us?
Yep. That's what it's going to look like when I show you. So that picture is an excellent choice to study. Okay, so check out the next page, page 103. So again, the testis is the organ, and then there's a couple parts. There are these big things that look like donuts, and the center of the donuts you can see little wispy tails. Do you guys see those little wispy tails? What are those? Sperm. So what would be the name of this organ? Testes. What would be the whole donut-shaped structure? Seminiferous tubules. So everybody check out how men make their sperm in tubes. Do women make their eggs in tubes? No, we make them in solid organs. Our ovaries are solid organs. But testes have tubes inside of them. They're tubular. Okay, and then in this picture, guys, there are little cells that are in between. Now, I don't know if you can see this far, but look up here, and I want you to find the big seminiferous tubule in the picture, and then go to the right towards the bottom. There's a little triangular piece of cells there. It looks like a little triangle. That is what I would use to show you interstitial cells. They labeled interstitial cells up above on this really tiny triangle. I'll use the big triangle. That's a better picture of interstitial cells. So if you want to draw that line down to the bigger triangle, that would be good. So, seminiferous tubules make sperm. Shake your head. Yes, they do. What do interstitial cells make? Excellent, testosterone. So, ladies, our ovaries make eggs. And what else? Estrogen. And gentlemen, your testes make sperm. And what else? Testosterone. Is everybody okay with that? Okay, so you need to know the stages of meiosis with the pop bead chromosomes. I got a couple vocabulary words. Synapsis. And this is number 6. What is synapsis? Synapsis. Anybody remember what's going on when we use the word synapsis? Yeah, Katie Lightfoot's right. The homologous chromosomes come together and they're like all twisty around each other. So I'll write that down for you. I'm just going to put H chromosomes. Is that okay with you?
Did we skip number 5?
No, you have to know the stages of meiosis with the pop bead chromosomes. I didn't skip it. I mean, you can study that. What page is that again? 96, 97? The stages of meiosis? Yeah. So synapsis would be when the homologous chromosomes come together and they twist around each other. So they get all intimate with each other.
The pop bead pictures on 93.
Yeah, how to build the pop beads is on page 93, and then the stages of meiosis with the chromosomes, page 96 and 97. I think that's correct. I have synapsis is when H chromosomes come together and wrap around each other. Check out page 96 and 97 and tell me, when does synapsis occur? Thank you, Alex. Prophase 1. Everybody check it out. Synapsis happens in prophase 1, the very beginning of meiosis. Okay, what is crossing over? They trade. What do they trade? Crossing over, what do they do? Yeah, they're going to switch pieces of DNA, so they're going to switch some traits. So we got the switching of DNA between homologous chromosomes. Why do they do that? Get you variation. Is okay on that one? Crossing over gets you variation. How can you look at a pop bead chromosome and know if it's done crossing over?
There's different colors.
Different colors. So the red chromosomes will have a little bit of, yeah, and the blue ones would have a little bit of red. Does crossing over occur in mitosis? No. Good. Okay. Mendelian genetics. Guys, this is a vocabulary list that I'm very sure you can find at the very end of your notes from last Thursday. So I'm going to move out. We're going to just leave that because you can look that one up. But I do want to answer a couple questions here before we go. What's the expected phenotypic ratio of a monohybrid cross? This is new for us, guys. A monohybrid cross looks like this. So let me explain why they call that a monohybrid cross. What does mono mean? One. So we're doing one trait. Thus you'll notice I only used one letter, the letter A. So you're just doing one trait. Hybrid means each parent is heterozygous. What does heterozygous mean? Hetero means the same or different? Different. So if you'll notice, this parent has two different genes for that trait. And this parent has two different genes for that trait. Now I'm looking for the phenotypic ratio, which is what they look like. And so they're either going to be dominant or they're going to be recessive. Does anybody know the answer? It is 3-to-1. What would that be in percents? 75%, 25%. You might like ratios, you might like percents. So I just want to be flexible there. So the monohybrid cross will always get you a 3-to-1 ratio, 3-to-1. But look at the second part of number 2. If you did a monohybrid cross of corn, would you expect the observed results to be closer to the expected ratio if you did 5 corn crosses or 50 corn crosses? 50, I agree. Why is it 50? Larger sample size. Are larger sample sizes good in science? Yes, it cuts down on randomness. You recall last week I kept making you flip pennies, nickels, and count corn? Do you remember we always came up to the board and put it on the board? And we always used all of our data because that way we had a big sample size. And I knew it would make us have better chi-square values. Big sample size is good in science. It cuts down on randomness. Today, okay, on the second part of number 2. Okay, check this one out. Dihybrid cross. Why is this a di? 2 traits. How do you know it's 2 traits? Because I used 2 different letters. Why is this a hybrid?
Because you're taking 2 different traits, or 2 different,
Use your words. Starts with an H. Heterozygous. Both parents are heterozygous. Heterozygous means hybrid. Hybrid means heterozygous. You guys know that word hybrid, like cars that are hybrid. What does that mean if a car is hybrid? It's 2 different energy sources. So it means different. Hybrid means different. Okay, does anybody know this ratio? So we did it up on this board right here last week.
9331.
It is 9331. Mm-hmm. If you go back to your notes in lab from last week, it's problem number 3 and problem number 4 from your notes from last week. I think we did nickels and pennies. Nickels and pennies. Okay, the last one I have time to do today in lab is what is the law of independent assortment? Okay, well, I'm going to let you look that one up. I'm just going to tell you that it is the random way chromosomes line up in metaphase 1. I think you can remember that the chromosomes line up randomly in metaphase 1. They don't care how they line up in metaphase 1 as long as they're across from their homologous partner. So I'm putting the random way homologous chromosomes line up, and it is in metaphase 1 of meiosis. But the most important part of this question is the second part of this question, so I'm going to star it. Does anybody know what the law of independent assortment gets me? Thank you very much, Emily. It's variation. It yields more variation in the cells. It gets you variation. Gets you variation. Okay, guys, downstairs I'll work on the chi-square stuff. I'll just do it for everybody. So make sure you have your calculator and your lab manual ready for lecture downstairs. And I will put this on YouTube. If you want to watch this review again, it'll be on YouTube.